Live from the ASBMR Symposium, Cutting-Edge Discoveries in Muscle Biology, Disease and Therapeutics, ASBMR President Linda Bonewald and Program Co-Chairs Karen Esser and Kevin Connolly discuss the most exciting science.
So there's a couple of questions I'd like to ask you, and they're really kind of personal and not scientific questions. And Karen, I'll start with you first. What's been your favorite session so far?
Well, I would say right now my favorite session is the first one today on the cells of muscle. Mm-hmm. I think all three of the speakers highlighted complementary but overlapping complexities to muscle. We've known about satellite cells and some of the stem cells in muscle for many years, but I think all three speakers, using some very elegant models and tools, really made quite a visual picture of the different cells that contribute to the muscle. Mm-hmm. And I think in terms of Andrew Brock's work, the fact that these things change over age and what they do. So as we think about aging, and again, we've thought about this for many years, but really teasing out their patterns of proliferation and activity at different stages of age, and how that may or may not contribute to some of the frailty and sarcopenia, I think is an exciting area. As well as coupled with, as someone said, the dancing satellite cells. So Dee Corneliussen's work.
The videos, right?
The videos were fun.
Okay. How about you, Kevin?
Well,
what was your favorite?
You know, I would move on now to I also found the first session to be just fascinating about both stem and satellite cells. But the second and third sessions got more into then diseases and their implications to muscle. And I found that particularly interesting. And not that diseases impact muscle, but they seem to impact muscle in many of the same ways through processes that call us sarcopenia, which is muscle wasting, mitochondrial dysfunction, insulin resistance. So there's a whole variety of things that impact the muscle, and they're remarkably in common with a whole variety of different diseases. So I thought that was very impressive.
Okay. So now, for me, there were a lot of surprises, and I learned a lot listening to the sessions. But I have to ask you, were there any surprises that you learned about today in any of the presentations?
Surprises? Well, I think Dee's observation that a cell divided within 4 hours, I think that's sort of outside what I'm familiar with.
That was a surprise.
That was a surprise. Mm-hmm. And the distance that some of those satellite cells were traveling.
It's phenomenal.
The rate was just really impressive. Mm-hmm. I think those were the really big surprises to me.
Any surprises for you, Kevin?
Well, and I thought one of the really big surprises was the level of sophistication in understanding some of the underlying mechanisms. And I was really impressed by things that were observed now are understood. And Daryl Neufert in particular, with his studies on insulin resistance and mitochondrial function, the level at which he could put mitochondrial function in terms of basic physical and chemical terms, and actually express mitochondrial function essentially as a proton and electron circuit, was, well, not only a wonderful teaching tool, but was really illuminating in terms of how these organelles work and how they go wrong in disease.
Mm-hmm. Mm-hmm. So my next question has to do with what's the hottest buzz as far as what's cutting edge that you know about, and did it come out in this meeting?
Well, I thought some of the work on the stem cells, again, I'm not very familiar with them, and when I saw the level of sophistication of what they can do with these cells that eventually become new muscle cells and regenerate, that was extremely impressive. And some of the in vitro work in which the stem cells moved and were motile and had some selectivity in terms of what cells they went to, I thought that's just for me a huge leap forward technologically in terms of our understanding, and for me it just gives me a lot of insight into how the whole system works. Mm-hmm.
And I would say too that the development of the non-invasive imaging technique. So I've sort of been in the basic sciences for a while. I've been watching the evolution of some of the work that Kevin mentioned, but I think, you know, in terms of translational research, the need for these fantastic new imaging tools and the work Kevin talked about, as well as Bruce Damon,
Right.
really are pushing the envelope in our ability to really bring bench to bedside.
Right.
Access the latest in bone education online, including these highlights from the ASBMR Symposium, Cutting Edge Discoveries in Muscle Biology, Disease, and Therapeutics, in the ASBMR Education Resource Center at www.asbmr.org.